A very good morning. I'm ALFARIZAL KAMARUDIN. Will be presenting my research entitled Chrysin and lentinan chemosensitized temozolomide in GBM. My research team at BRINT is focusing on glioma, particularly glioblastoma or GBM, which is known to be incurable. Although the standard therapy of GBM may extend the patient's survival up to 15 months, the GBM tumor's nature, such as high heterogeneity, often confers therapy resistance that contribute to the therapy pitfall. These often then lead to a low overall and progression-free survival with a high rate of clinical recurrence. As an attempt to overcome these challenges, we would like to employ the use of phytochemicals which are known to be multi-target with marginal toxicity while capable of acting as adjuvant to the existing therapy. We employ the use of chrysin, a flavonoid commonly found in honey and propolis, and lentinan, a polysaccharide that can be found from shiitake mushroom, since they are both capable of multi-targeting various signaling in cancer model. Taking this into consideration, their combination with temozolomide was hypothesized to synergize temozolomide-induced apoptotic cell death in human GBM. Moreover, they have never been studied in chemo-resistant human GBM models, although their combination with other chemo drugs are well reported in other cancer models. While chrysin is less studied in chemo-resistant GBM and lentinan has never been studied in human GBM, their combination with TMZ have never been reported. Thus, by achieving the specific listed objective, both chrysin and lentinan may be developed as potential nutraceutical-based adjuvant in human chemo-resistant GBM. To achieve the objective, we employed the use of two different GBM cell lines, LN18 and T98G, which were subjected to single treatment of chrysin, lentinan, or temozolomide in MTT-assay. Following the IC50 determination, the synergistic effect of chrysin and lentinan in combination with temozolomide was conducted using isobolographic analysis, which was then followed by the cytometric apoptosis study and elucidation of key protein using just simple Western. In our analysis, both chrysin and lentinan significantly reduced the GBM cell viability more efficiently than temozolomide. Chrysin effect was more pronounced in the LN18 cell, while lentinan was more pronounced in the T98G cell. The isobolographic analysis further demonstrated that chrysin addition to temozolomide resulted in a good synergism, while lentinan demonstrated a strong synergism. We then chose 3 best CI value for each cell line for the apoptosis studies. In our preliminary analysis, chrysin at 30 micromolar, while lentinan at 50 micromolar promoted the best apoptotic effect compared to the other concentration. Thus, this concentration was then selected for the subsequent assays. Our data demonstrate that temozolomide combination with chrysin or lentinan promote the apoptotic cell death higher than the single drug treatment despite the lower temozolomide concentration. In order to validate the intrinsic apoptosis, we conducted mitochondrial membrane potential assay. The current data demonstrate that a combination treatment of temozolomide with chrysin further promotes the mitochondrial membrane depolarization that induces apoptosis, while lentinan addition resulted in a slight increase of the depolarized cell. The promotion of apoptosis is generally regulated by the interplay between pro-apoptotic and anti-apoptotic protein level, leading to caspase activation. Our data demonstrate that the addition of chrysin into temozolomide treatment significantly enhanced the reduction of anti-apoptotic protein. Meanwhile, the same treatment paradigm increased the pro-apoptotic protein level, leading to a more favorable apoptosis condition in the LN18 cell. Following this observation, chrysin with temozolomide further reduced pro-caspase-9 entry level compared to the single drug treatment. Additionally, this was preceded by the inactivation of AKT and ERK12, but the activation of P38 MAPE kinase compared to the single drug treatment. Meanwhile, lentinan addition significantly reduced the BCL-XL but increased the back protein level, which led to the activation of caspase-9 and 3. However, this part of study is still incomplete and ongoing due to the MCO interruption. To corroborate the involvement of extrinsic and intrinsic apoptosis, the cell were then pretreated with caspase inhibitor, where caspase 9 and 3 addition significantly suppress apoptosis with sustained level of pro-caspase 9 and 3. Additionally, pretreatment with either API2 AKT inhibitor or UO126 ERK12 inhibitor further promoted apoptosis in the LN18 cell. In contrast, pretreatment with the SB2021NO P38 PEP kinase inhibitor significantly suppressed apoptosis, further indicating that the activation of P38 MAPE kinase and inactivation of AKT and ERK indeed are required for the apoptosis promotion by Chrysin and temozolomide combination in the LN18 cell. Collectively, both Chrysin and lentinan possess the ability to promote temozolomide cell death via a caspase-dependent apoptosis in chemo-resistant GBM cell, even at lower concentration. In essence, our current data support the combination use of Chrysin and lentinan at lower concentration with TMZ, indicating their potential as nutraceutical-based adjuvant in improving the chemo drug efficacy. Toward the sustainability of this research, this project will further elucidate the apoptosis mechanism and induction of ferroptosis through an innovative delivery system while targeting novel target in GBM that move from the direction of preclinical to clinical therapeutic in GBM. Thank you for listening, and my sincere gratitude to all parties that have contributed to this project at BRIMS.